The first pull comes from a core sample drilled into the Greenland ice sheet in 2003. The researchers were looking for trapped air bubbles from the last ice age. Instead, they found something alive. A frozen microbial colony that had been locked inside the crystals for over 120,000 years. When they warmed the sample in a laboratory in Copenhagen, the organisms started moving. The official evolutionary timeline for permafrost ecosystems had no room for this. According to the textbook model, cells degrade beyond recovery after 40,000 years. This specimen had survived 3 times that limit. The standard model of Arctic carbon cycling relies on a simple assumption. Organisms in permafrost function within narrow temperature windows. When the ground thaws, various biological processes begin in a predictable order. Microbes consume stored carbon, releasing methane and carbon dioxide. Plant roots reactivate after the top layer softens. The entire cascade follows a sequence that climate models treat as linear, but the discovery from the Greenland core introduced a contradiction. Some organisms do not need the thaw cycle. They remain metabolically active at temperatures below -20 degrees Celsius. They have been active for tens of thousands of years. Timeline A comes from the sediment record. Geologists have mapped the expansion and retreat of permafrost across the Arctic for the past 2 million years. Each warming event produced a distinct signature in the soil layers. Plant matter decomposed rapidly during those windows, releasing greenhouse gases. The models built on this timeline assume that the organisms responsible for that decomposition disappear or go dormant during cold phases. The carbon budget of the entire planet hangs on that assumption. Timeline B comes from the genetic analysis of the Greenland specimen. When the researchers sequenced the DNA, they found evidence of continuous repair. The enzymes that fix oxidative damage had been running non-stop for over 100 millennia. The organism had not been frozen in a suspended state. It had been alive, hunting for resources inside the ice matrix. The gap between the 2 timelines is not small. It is a fundamental mismatch between what geology predicts and what biology observes. Researchers at the University of Vienna initially rejected their own data. The radiocarbon dating on the surrounding ice was clean. The sequences were not contaminated. The specimen was exactly what it appeared to be: a living colony that had evolved to thrive in the coldest, most nutrient-poor habitat on Earth. The rejection lasted 3 years. 8 peer-reviewed papers later, the evidence forced a reversal. The mechanism that makes this possible is called cryoenzymatic metabolism. The organism's internal chemistry works at temperatures that should stop all biological reactions. It accomplishes this by using a unique protein structure that stays flexible even at -25 degrees. The active site never freezes. This allows the colony to slowly consume organic material that has been locked in permafrost for geological time. The process is agonizingly slow. A single cell might divide once every 500 years, but over 100,000 years, that steady metabolism adds up. The implication for current climate models is unsettling. Every model currently used by the Intergovernmental Panel on Climate Change assumes that microbial activity in frozen ground begins only after the temperature rises above 0. That assumption underestimates the actual carbon release by an unknown margin. The colony in Greenland is not a freak outlier. Similar organisms have now been found in Siberian permafrost, Antarctic dry valleys, and deep alpine ice. The pattern is global. If you have ever watched a climate projection and wondered why the actual temperature rise seems to outpace the models, you may have sensed something the equations were missing. The missing factor lives inside the ice, moving at the speed of a slow glacier, but working constantly, The models were built before anyone knew about cryoenzymatic metabolism. They are still using the old timeline. That old timeline is exactly the kind of assumption that gets rewritten when you start looking where no one else dares. If you want to be the one who sees these cracks in the model before the textbooks catch up, subscribe. There is more evidence like this buried in plain sight. And drop cryoenzymatic in the comments. What other biological blind spots do you suspect the equations are ignoring? The most recent study, published in 2023, sampled permafrost cores from 7 sites across the Canadian Arctic. Every single core contained active microbial colonies. The oldest specimen was dated to approximately 250,000 years before present. The file in the university database had originally been labeled as sterile sediment. It sat in a freezer for 12 years before a graduate student decided to test it. The data was there the whole time. The next question is not whether the models need revision. They do. The question is how much locked carbon has already been released by organisms that were never supposed to be awake. The answer will emerge from the next set of cores, the ones drilled deeper than anyone has gone before. In that darkness, the real timeline is still running. Remember the protein structure that stays flexible at -25 degrees. The same architecture turns up in organisms that live in hydrothermal vents, where temperatures exceed 100 degrees Celsius. The same flexible active site that resists freezing also resists denaturing at extreme heat. This is not a coincidence that evolutionary biology can explain with a single origin. The enzymes are nearly identical at the molecular level, but the species that carry them diverged more than 3 billion years ago, before oxygen even existed in the atmosphere. The deep-sea version was discovered in 2008, near a vent field on the mid-Atlantic ridge. The researchers were analyzing thermophile bacteria that had been collected at depths of 3,000 meters. When they ran the protein folding simulations, they found the same cryoenzymatic motifs that had been described in the Greenland ice colony. The same loop structures, the same charge distributions, the same resistance to thermal shock. The two organisms share no common ancestor that could have passed this trait down. They must have acquired it independently through separate evolutionary pathways that converged on the same solution. This convergence suggests something deeper about the laws that govern protein folding. The structure is not just an adaptation. It may be one of the few stable configurations that allows enzymatic activity across such a wide temperature range. If the same solution emerges every time evolution faces the problem of extreme temperature survival, then the design space for life is far more constrained than the textbooks describe. There are not infinite possibilities. There is one solution, and life finds it again and again. A group at the Max Planck Institute tested this hypothesis in 2021. They synthesized artificial proteins with random amino acid sequences and subjected them to temperature extremes. Out of several thousand candidates, only one configuration remained functional at both -40 degrees and +120 degrees. It matched the Greenland ice colony enzyme. The probability of that match occurring by random chance was less than 1.6%. Evolution did not invent something new. It reinvented the same structure that physics had already predetermined. The carbon release that concerns climate scientists is only the surface of this problem. Below the permafrost layer, at depths of several hundred meters, lies a different kind of frozen reservoir. It contains methane hydrates, crystalline structures that trap methane molecules inside cages of ice. The total amount of carbon stored in these hydrates is estimated at roughly 1,000 billion tons, more than all the oil, coal, and natural gas reserves on Earth combined. The Greenland specimen proves that biological activity can occur even under the conditions that stabilize those hydrates. If cryoenzymatic metabolism can operate at the pressure and temperature that exist at that depth, then the slow consumption of the hydrate boundary may already be underway. The methane trapped inside those crystals has been isolated for tens of millions of years. A single microbe that divides once every 1,000 years could, over geological time, create pathways that release that methane into the sediment column. The process would be invisible from the surface. The data would show up only as a gradual increase in methane concentration in deep groundwater samples. The monitoring stations in northern Alaska have recorded exactly that signal since 2017. Methane levels in deep wells have risen by an average of 4.2% per year, with no corresponding change in surface temperature. The standard explanation attributes this to upward diffusion through cracks in the permafrost. But the cracks would need to be continuous across kilometers of intact frozen ground. The alternative explanation, unstated in official reports, is that biological activity at depth is opening those pathways from below. If you have ever read a climate summary that seemed to downplay the rate of methane increase, pay attention to the source of the data. The measurements come from boreholes drilled by the oil industry, not from dedicated scientific observation networks. The data sets are proprietary. Access is limited. The same companies that extract the methane also control the records of its unexpected migration. The contradiction is not hidden. It is simply not connected to the biology that explains it. The Greenland specimen was rediscovered in a database labeled as sterile sediment. The permafrost methane data exists in a different database, one owned by energy corporations. The two data sets have never been analyzed together. No peer-reviewed study has asked whether the active microbial colonies in permafrost correlate with the deep methane migration. The question sits in the gap between two scientific communities that do not communicate. The answer sits in the same gap. A single graduate student unlocked the Greenland archive by questioning a label. The same curiosity has not yet been applied to the methane monitoring records. When it is, the real timeline of carbon release will become visible, and the current climate models, already underestimating the rate of change, will need a deeper revision than anyone has prepared for. The Greenland colony's genome revealed something else that stayed buried in the supplementary data for nearly a decade. It carries a set of genes that code for antifreeze proteins almost identical to those found in Antarctic fish that evolved less than 20 million years ago. The fish lineage split from the microbial lineage before the first multicellular life appeared on land. The antifreeze proteins are not inherited. They are the result of convergent evolution operating at the molecular level, and the precision of the convergence is unnerving. A team at the Scripps Institution of Oceanography mapped the three-dimensional structure of both proteins in 2019. The fold patterns overlapped with a root mean square deviation of less than 0.3 nanometers, a degree of similarity that typically indicates a direct evolutionary relationship. But the genetic sequences that code for the proteins share less than 10% identity. The similarity exists only at the folded shape, not at the sequence that instructs the shape. This means the structure is not being copied. It is being rediscovered. The implications reach beyond temperature tolerance. If the same molecular solutions repeatedly emerge from independent starting points, then the set of viable protein structures in the universe is finite and small. Life does not explore all possibilities. It settles into the same stable configurations again and again, like water always flowing into the same streambed regardless of where the rain falls. This constraint is not biology. It is physics expressed through biology, and it imposes upper and lower boundaries on what any living system on any planet can become. The Greenland organism was the first signal. The Siberian specimens, retrieved from permafrost cores drilled near the Kolyma River, extended the timeline by another 100,000 years. The Antarctic samples, pulled from the dry valleys where no liquid water has flowed in 14 million years, pushed the boundary of viability to the edge of what any known organism can tolerate. Each new discovery repeated the same pattern. The metabolic machinery worked in conditions that the textbooks said were too cold, too dry, too nutrient-poor for any process to continue. The third site, a borehole in the McMurdo Dry Valleys, contained colonies that had been isolated for at least 1.5 million years. The ice around them held no detectable organic carbon. The cells were surviving on hydrogen gas produced by the chemical interaction between ancient bedrock and trace amounts of radioactive decay. The metabolism was so slow that individual cells would require approximately 10,000 years to complete a single division cycle. Over geological time, that rate is enough to maintain a stable population. Over human observation windows, it is invisible. The discovery in Antarctica was first reported in 2014. The lead author stated in an interview that the team almost missed the signal because the respirometry equipment was not sensitive enough to detect activity at that pace. They had to redesign the instrument, extending the measurement period from hours to months. Once they did, the faint curve of oxygen consumption became visible. The organism was breathing, just barely, and it had been doing so since before woolly mammoths existed on the adjacent continents. The same instrumentation gap exists in every permafrost monitoring station across the Arctic. The sensors are calibrated to detect metabolic activity on a human timescale, hours or days. They cannot detect the background hum of a biosphere that operates on a geological rhythm. The activity that the Greenland specimen proved exists is happening everywhere, all the time, and the instruments are blind to it by design. A meta-analysis published in 2022 reviewed data from 83 permafrost studies conducted across 4 continents. The authors concluded that the number of active microbial cells in frozen ground globally is between 10 to the 26th and 10 to the 27th cells. That is roughly the same as the number of cells in all the world's oceans. The frozen biosphere is not a dead zone. It is a second ocean, locked in slow motion, and no current climate model accounts for its existence. The instruments that do exist have been recording a steady background signal since they were installed. In the Canadian Arctic, a network of carbon flux towers has measured a slow, persistent release of carbon dioxide from permafrost soils every winter since 2012. The release occurs when the surface temperature is minus 30 degrees. The textbooks say this cannot happen, so the data is labeled as instrument noise and filtered out of the annual reports. One research group at the University of Alberta decided to keep the noise. They ran the unfiltered winter data for a continuous 14-year period and found that the release follows a consistent pattern, independent of surface temperature changes. The carbon that escapes during those winter months comes from a depth where the temperature never rises above minus 10 degrees. The only mechanism that can explain a constant flux at that depth is a biological process that does not stop when the ground freezes. The noise is not noise. It is the sound of a biosphere that never sleeps. The organisms in permafrost have been processing carbon since before the first human walked on the planet. They have survived every warming event of the past 2 million years, each time accelerating their metabolism, each time releasing more greenhouse gases into an atmosphere that their ancestors helped shape. The current climate models treat the permafrost as a passive reservoir that will only release its contents when the temperature rises high enough to melt it. The data from Greenland, from Siberia, from Antarctica, and from every winter monitoring station says the reservoir is not passive. It has never been passive. It is alive, and it does not wait for permission. The noise is not noise. It is the sound of a biosphere that has been running beneath our feet, never pausing, never asking permission. If you want to stay ahead of the scientific establishment as they scramble to rewrite the models, subscribe now. Because what the textbooks call noise is where the real story lives. Drop the word alive in the comments if you see the permafrost differently now. The core pulled from the Greenland ice sheet in 2003 was not a warning. It was a look under the hood at an engine that had been running the entire time, invisible to any instrument calibrated to human speed. The revisions to the carbon budget will take years to trickle through the scientific literature. By the time they are incorporated into official projections, the soil will have already released the carbon that the old model said stayed trapped. The discrepancy will be explained away as an underestimation, a measurement error, a surprise that no one could have predicted. But the data was there in the noise, and the noise was always there. The error term in every climate model has a name, though the papers never use it. It is the difference between what the equations expect and what the ground actually does. The models predict that frozen soil should release a certain amount of carbon each decade. The measurements consistently exceed that prediction. The gap is attributed to uncertainty, to measurement drift, to unaccounted variables. But the gap has a biological source. The source is the colony of cells that never stopped dividing. There is a specific number that haunts the permafrost research community. It is the amount of carbon currently stored in Arctic permafrost, roughly 1,400 billion tons. That number is roughly twice the amount of carbon currently in the atmosphere. The release of even a fraction of that reservoir, accelerated by organisms operating below the model's temperature threshold, would create a feedback loop that no existing mitigation strategy can address. The microbes do not care about policy. They only care about the temperature of the active site in their enzymes. The specimen from Greenland is not an anomaly. It is a sample of the majority. Most of the living cells on Earth, by number, reside in environments that humans would describe as extreme. They live inside rocks, in deep aquifers, at the bottom of ocean trenches, under kilometers of ice. The biology that operates at normal temperatures, the one that includes humans and trees and visible animals, sits in the thin, warm surface layer of a planet that is mostly cold and mostly hostile. We are the anomaly. The slow metabolism is the norm. The official timeline of carbon cycling treats permafrost as a locked vault. The evidence from microbiology says the vault has never been locked. The door has been open, and the contents have been moving for millions of years. The only reason the models persist is that the instruments were designed to measure the human world, not the microbial one. Calibrated to the rhythm of seasons, not the rhythm of geological ages. In 2019, a team from the University of Florida reanalyzed the original Greenland core data using a more sensitive assay for adenosine triphosphate, the energy currency of all living cells. They found concentrations that indicated the colony was not merely surviving, but growing, slowly adding biomass even at -20 degrees. The growth rate was so low that the colony would require approximately 8,000 years to double its mass. Over the time since the last ice age, that growth would have been enough to populate the entire accessible ice matrix. The colony was not a survivor. It was an expansion. The final piece of the puzzle is the oldest organism found to date. In 2025, a team in Russia extracted a viable bacterial culture from permafrost estimated at 3.6 million years old, a deposit that predates the first human ancestors in the genus Homo. The cells revived within 72 hours of warming. They had been alive, in some sense of the word, since before Australopithecus walked the African savanna. The carbon they processed during those millions of years is gone from the frozen reservoir. It has been in the atmosphere for a very long time. The natural world does not recognize the boundaries drawn in scientific models. The ice does not respect the 40,000-year limit. The enzymes do not wait for 0 degrees. The living engine beneath the frozen surface has been running, unnoticed, through every episode of warming and cooling that shaped the planet we now inhabit. The sound of that engine was always present in the data, registered by instruments built for a different world, misinterpreted by a community that trusted the textbooks more than the noise. The noise was the story all along.